Hi dear friends, in this video I would like to explain major enzymes and proteins that's involved in DNA replication. Okay, what are the enzymes and proteins that is involved in DNA replication? Okay, first one is a DNA helicase. Okay, DNA helicase is an enzyme that's break hydrogen bond between the two DNA strands during replication. This DNA helicase enzyme helps to remove hydrogen bonds between the DNA strands. And the second one is a DNA topoisomerase. And the second one is DNA topoisomerase, which alleviates or remove DNA supercoiling, that means DNA positive supercoiling, and DNA topoisomerase is present in the eukaryotic cells, and another one enzyme that's present that gives same procedure as as DNA topoisomerase, that is in bacteria, that is in DNA gyrase enzyme. DNA gyrase is like DNA topoisomerase is DNA gyrase is function like DNA topoisomerase 2 in eukaryotic cell, which remove positive supercoiling. And another one protein involved in DNA replication is SSB, that is single strand binding protein. Okay, single strand binding protein. This protein helps to keep the parallel strand apart. Okay. During the replication of DNA, these two DNA strands will be kept apart. That this single strand binding protein will keep this two DNA strands apart, and this single strand binding protein also protect the DNA strand. Okay, and this single strand binding protein also protect the DNA strand from some nucleases. Okay, and another one enzyme, that is a PRIMASE. PRIMASE is an enzyme that used to synthesize an RNA primer during the process of replication. RNA primase is an enzyme that synthesizes an RNA primer during the time of replication. Okay, synthesize RNA primer during the time of replication, and primase is an enzyme. RNA primase is an enzyme that is an RNA polymerase. And another one is a DNA polymerase. Synthesize a double strand of DNA in 5' to 3' . Okay, this DNA polymerase is an enzyme that is synthesized daughter strand of DNA in 5' to 3'. And in eukaryotics, there are mainly five type of polymerases. One is alpha, beta, delta, gamma, epsilon. Okay, alpha, beta, gamma, delta, epsilon. There's a eukaryotic polymerase present. And another one is prokaryotic polymerase, that is bacterial polymerase, that is DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. And another one enzyme that is involved in DNA replication is a ligase. Ligase is an enzyme that will help to link the Okazaki fragment together. In DNA replication, Okazaki fragments are ligated by this method, and this also helps to link the DNA, fill the DNA, seal the DNA gap. These are the enzymes and protein that's present in the replication of DNA. And one is the DNA helicase. DNA helicase break the hydrogen bond between the DNA strands. And the other one is the DNA topoisomerase is elevates positive supercoiling. And there is two type of DNA topoisomerase present in eukaryotic. That's DNA topoisomerase 1 and 2. And in the case of bacteria, there is an enzyme DNA gyrase that function like DNA topoisomerase 2 in bacteria. And another one protein is a single strand binding protein SSB that keep the DNA strands apart. okay, that keep the DNA strand apart during the time of replication, and then also this parallel strand away from protect the DNA strand from the nucleases. And another one enzyme, the PRIMASE, which synthesize RNA primer during the time of replication, and this RNA primer is an RNA polymer. Okay, RNA primer is there, enzyme is an RNA polymer. Another one important is a DNA polymerase. DNA polymerase synthesizes double strand of DNA in 5' to 3' direction. And eukaryotes contain there is 5 types of DNA polymerase. One is alpha, beta, gamma, delta, epsilon. And in the prokaryote there is a 3 types of polymerase only present. That is polymerase 1, 2, 3. And another one enzyme is a DNA ligase, is also known as molecular glue, and which covalently links the active fragment together. Okay, helps them link fragments of DNA in replication. These are the major enzymes and the proteins involved in DNA replication. I hope you understood. And in this figure, you can see the presence of this each enzyme in replication form. I hope you understood this class. Thank you for listening.